Hi guys, welcome to today's lecture. So I hope you guys recall that last time we spoke a bit about non-specific host defense. Well, today we'll do part one of two lectures on specific defense of the host or immunity. Um, so just a specific defense is also known as adaptive immunity, and it's called this because the first time you respond, your immune response will be slow and not very strong, but afterwards your immune system will be altered to this pathogen, and subsequent responses thus will be much stronger. So in a way, your body has adapted to fight this pathogen and won't be affected as negatively unless the pathogen, of course, changes. So how do some characteristics we should know here is, so specific defense or adaptive immunity refers to defenses that target a specific pathogen. Very important here that we will start dealing with specific pathogens, and these aren't catch-all solutions as we discussed previously. Um, we acquire this type of immunity through infection or vaccination. Very important now with the entire COVID thing. I hope you guys are all going to go get your shots. Very important. And then, so the primary response is what happens the first time the immune system combats a particular foreign substance. Again, this response will typically take much longer and won't be as strong. The secondary response to the same foreign infection, to the same pathogen, will be much stronger due to essentially the memory that you have obtained and your how you've adapted to it. So yeah, that's the difference between those two. So in the adaptive immune system, you will find that there are two predominant mechanisms. There's humoral immunity that is driven by your B cells, and then there is also cell-mediated or cellular immunity that's driven by your T cells. So humoral immunity is where you produce antibodies, and they typically defend against bacteria, bacterial toxins, and viruses. But importantly, they defend against these agents when they are circulating freely in the body fluid. So they are not inside cells just yet. Where with cellular immunity, they will also act against bacteria, viruses, but this occurs within the host cells. Also, fungi, protozoa, and helminths are also combated by your T cells. So, essentially, some parasitic organisms. And they can also provide some defense against foreign tissues such as cancer. So those are some important distinctions. Today, we will be most heavily focusing on the B cells, and then the part two of the lectures will be looking at the T cells. So both types actually start out as stem cells in the bone marrow. So that's what's represented over here. This can also be called a progenitor lymphoid cell, is what you start off with. And then when you have B cells, somewhere that you can remember the B for B cells because of bone marrow. So essentially, the cell will then move to the bone marrow where it will differentiate and then become a B cell. Where conversely, on the other side, if this progenitor lymphoid cell goes to the thymus, it'll differentiate to T cells within the thymus. And then once these cells are formed, they will migrate to lymphoid tissue such as the spleen and especially obviously lymph nodes. So you can also remember the T cells, remember the T in the thymus and the B for the bone marrow. It's just an easy way to remember where these cells then come from. Okay, so again, just as a reminder, B cells attack outside the cells, T cells attack inside the cells. So moving on to antigens. So humoral immunity is the aspect of the immunity that is mediated by macromolecules found on the extracellular fluids, such as secreted antibodies, complement proteins, and certain antimicrobial peptides. Humoral immunity has that name because it involves substances found in the humors, so body fluids. And now we will move on to antigens, because here it's important that antigen, actually this word stems from antibody generator. So an antigen is just simply a molecule capable of inducing an immune response on the part of the host organism. It's important to know that sometimes antigens can be part of the host itself. So in other words, an antigen is any substance that causes an immune system to produce antibodies against it. Antigens often include components of the capsule, cell wall, flagella, or fimbriae, comprising proteins and large polysaccharides. You also have haptens and carrier molecules here. Haptens are just small molecules that elicit an immune response only when attached to a larger carrier molecule, such as a protein. So essentially, the hapten is an incomplete molecule. It does need its carrier molecule, and thus it will produce a complete antigen. So these haptens will not produce an immune response by themselves. They need the carrier molecules. And then epitomes, just the definition that I want you guys to be aware of, that's the region on the antigen that the antibody will interact with. So this is where your recognition and attachment will occur. So moving on to just giving you guys a brief overview here we have a bacterial cell. We have some binding sites on both the antibody, represented by this green Y, and on the bacterial cell. And here you can see as an example of some epitomes. So this is where the attachment will occur. So generally, antibodies recognize and interact with specific regions on antigens called these epitomes or antigenic determinants. The nature of this interaction will depend on the size, shape, and the chemical structure of the binding site on the antibody molecule. So again, just in this illustration, the epitomes are the components of the antigen and the bacterial cell wall. Each antigen carries more than one epitome, and each Y-shaped antibody molecule has two binding sites. So there's a binding site over there and over there. So this antibody is already binded to it, and there you can see both binding sites. And each of them will attach to a specific epitome of an antigen. An antibody can also bind to identical epitomes on two different cells at the same time, which can cause neighboring cells to aggregate. We will be looking at that a bit more later on, so don't worry about that too much right now. So most antigens have a molecular weight of 10,000 or higher. So a foreign substance that has a low molecular weight is often not antigenic unless it is attached to a carrier molecule. Again, here we're going to be looking at these haptens. So here you will see the hapten molecules, the smaller molecules, and in this form, they will not elicit an immune system response. They need to be associated with the carrier molecule, which will then make this hapten carrier conjugate, and now it will actually produce an immune response. So just a bit of background here. I often found studying that if I know the origin of words, they are easier to understand. So hapten actually originates from the Greek hapto, which means to grasp. So you can think of these little molecules need to grasp the carrier molecule in order to become active. So antigens need this larger size in order to actually be functional. However, once an immune system identifies haptens in the future, the antibodies, it can elicit an immune response on its own. So that's also an important thing that you should be aware of. So now we're going to be quickly discussing the composition of blood. Firstly, just looking at antibodies. So antibodies are globulin proteins. So this is a protein with a compact globular form. Therefore, we have come to use the term immunoglobulin, so IG, for antibodies. And antibodies are made in response to an antigen and can recognize and bind to an antigen. Just a reminder again there. Different antibodies are found in different tissues. We'll be looking at some more examples, but the most common ones is the IgG, which is found in blood and tissue. And thus, we're going to just do a quick overview of the composition of blood, as I said before. And I'd like to just remind you guys, in terms of our breakdown of the immune response and my breakdown that I'm going to give you guys about the blood here. This is an extremely simplified and basic breakdown. This is not a physiology class, so I'm not going to spend too much time dealing with the physiology of things. So this is just to get you to understand the immune response. It's very, very simplified. So I understand you will have there will be some physiology majors in the course that will obviously perhaps know more about this and will find this a little boring, but just bear with me here. So just getting back to our overview of blood. So blood will comprise of cellular material. This would be your erythrocytes, which are some blood cells or red blood cells, your leukocytes, which are your white blood cells, and then your platelets. And then the term plasma is just essentially referring to your blood minus the cells. So this will include your salts, soluble metabolites, and proteins. Your proteins here will be fibrin, albumin, complement, and globulins. Important that globulins, this is where our antibodies will be. Important also here that you know that fibrin is also called factor Ia, and it's a fibrous non-globular protein that is involved in the clotting of blood. Albumins, on the other hand, are a family of globular proteins, the most common of which are the serum albumins, and all the proteins in the albumin family are water-soluble, moderately soluble in concentrate and salt solutions, and experience heat denaturation. And now moving on to the globulins. So globulin or spheroproteins are spherical, so globe-like proteins, and they constitute a group of simple proteins soluble in salt solutions, forming a large fraction of the blood serum protein. And important here that the three principal subsets of the globulin are your alpha globulin, beta globulin, and gamma globulin, which are distinguished by the respective degree of electrophoretic mobility. So alpha has the greatest and gamma having the least. So what in the world am I talking about? So this is essentially what I was referring to just now, where you do a experiment to see your protein migration or the ability of your proteins to move once subjected to this electrical field, I suppose you can say. So essentially here, you will, if we move from left to right, here first off you'll find your gamma globulins, and they are found in your blood serum, but also other bodily fluids, particularly lymph system and mucous membranes. Moving on, then you will find your beta globulins, which are a group of globular proteins in the plasma that are more mobile in alkaline or electrically charged solutions than gamma globulins, but less than your alpha. So essentially you can see that by how they moved through this material once exposed to the electric charge. They moved from this trough. So you can see that based on how rapidly these proteins moved, you can differentiate between them. So we're at beta, moving on to alpha. So these are also a group of globular proteins in the plasma that are highly mobile in alkaline or electrically charged solutions, and they inhibit certain blood processes and show significant inhibitor activity. And then finally, we will have albumin, which is the most prevalent protein in blood, and obviously they show the greatest mobility. They play a role in regulating osmotic pressure. Okay, so blood distinguishing aside, moving back to the structure of antibodies and the antibody molecule. So you will see that I hope that you will recall, at least because I mentioned this earlier, that each antibody has at least two identical sites that bind to the epitomes. Here you can see the two sites on the ends here. These sites are obviously they're known as your antigen binding sites, and the number of antigen binding sites on an antibody is called the valence of that antibody. So, for example, most human antibodies have two binding sites. Therefore, they are bivalent. Because a bivalent antibody has the simplest molecular structure, it is called a monomer. And a typical antibody monomer has 4 proteins, 2 identical light chains, you can see in the blue, and then 2 identical heavy chains, represented by the top green. Here, the light and the heavy will refer to their relative molecular weights. And the chains are then joined by disulfide links and other bonds to form a Y-shaped molecule. Here you can see the disulfide links that link this entire antibody together. Please note, though, that however the Y-shaped molecule is flexible, it can also assume a T shape. So please notice the hinge region here, where you can still have some movement, so it can still flatten out to resemble a T shape. So then also what I want to point out here, notice the C. The C here stands for a constant region. There are 5 major types of constant regions, which account for the 5 major classes of immunoglobulins. Keep that in mind when I move on to the next slide. And then the 2 sections located at the end of the Y arms are called the variable regions. Here you can see them circled. Well, that one is circled at least. And here you can see it also represented by the V's. This is the one that I really want you guys to focus on, because again, these are the sites that bind to the epitomes, and the amino acid sequences, and therefore the 3-dimensional structure of these variable regions are identical on any one antibody. Their structure reflects the nature of the antigen for which they are specific, however, meaning that they are specific to the 2 antigen binding sites found on each antibody monomer, and they can shuffle genes around here to produce random variable regions. Very important. So there are lots of different pathogens, viruses out there. I'm sure you guys are getting a pretty good idea of that by this point. So we need to have a large variety of receptors, and that is how that is accomplished essentially. So remember the 5 different classes I mentioned. So they're, like I said, the simple and most abundant of the immunoglobulins are monomers, but they can assume differences in size and arrangement, and that produces the 5 classes. So first we'll have IgG. So this G at the end stands for gamma, because I find in gamma globulin fraction. Then IgM, M is for macro, because they are large. This class makes up 5 to 10% of the antibodies in serums. Then you find A. A is for attachment, because they prevent the attachment of microbes to cells in mucous membranes. D here is for don't know, so they have no well-defined function. And they're found in blood, lymph, and particularly on the surface of B cells. E you can remember by thinking they evoke allergy. So these are specialized cells that participate in allergic reactions, and they are found in serum. So just for memorization sake, G, you can think of gamma, M, macro, A, attachment, D, because we don't know, it's not well defined yet, E, and for evoke allergy. I would like to just ask you guys here, again, this is a very simplified breakdown, so please be sure to review this section in your textbook. I was very brief on the details. There's quite a bunch of fascinating facts in your textbook, and you can definitely get more insight there. Just in the time, in the effort to keep this lecture as concise as I can, I did skip out quite a few details, so please make sure to have a look at that. Now we also need to discuss the concept of immunological tolerance. So immunological tolerance is a tongue twister, A, and B, it is the failure to mount an immune response to an antigen. So this can be either natural or self-tolerance. So again, this is the failure, which is in this case a good thing, to attack the body's own proteins and other antigens. So if the immune system would respond to yourself, you would essentially have an autoimmune disease, which can be very, very devastating and even deadly. So under normal circumstances, if you don't have an autoimmune disease, you do have a tolerance to yourself, and that is where you find this immunological tolerance. A difficult word to say. Again, what I just thought I'd include this. This comes straight from your textbook, so you guys definitely want you guys to have a look at this. This is a very nice quick reference for exam purposes. I do want to point out one difference here. I want to look specifically at IgD and IgE. Note that they differ in the structure of their heavy chain, which is definitely something I want you guys to be aware of. But again, this is just a nice little uh, breakdown of the immunoglobulin classes and some of their factors. So very handy for exams. Okay, so now we're going to be start looking at the synthesis or the creation of antibodies. So just a brief breakdown here. So antibodies are produced by B cells. Very important. They're exclusively produced by B cells. They're not produced by T cells. Stem cells in bone marrow and the fetal liver. Some B cells can then migrate to the lymphoid organs. We showed that. Recall that diagram we had at the start. And then the antigens will then stimulate B cells to differentiate into the plasma cells, which actively secrete soluble antibodies. So what is our end result is we have an antigen-antibody complex that is formed that causes neutralization, opsonation, which we'll be discussing, agglutination, and more. So it just hampers these disease-causing microbes, essentially. Remember, at the end, we will also have some form of immunologic memory, meaning we will have a greater, a more effective response should we encounter the same pathogen again. And thus, so the formal definition here would be that this is the ability of the immune system to quickly and specifically recognize an antigen that the body has previously encountered and initiate a corresponding immune response. Again, these secondary, tertiary, and subsequent immune responses are typically much stronger than the initial one, so you don't get as sick. This is what we were just on about, is how they are differentiated and to the point where they get there. So again, we were focusing only on the B cell side here. And while, what I'd like just also to note that while these B cells are differentiating within the bone marrow, they're exposed to lots of different molecules, proteins, polysaccharides, that are produced within the body. So if a cell recognizes a molecule within the bone marrow, it is destroyed. And this is how our body essentially prevents making B cells that produce antibodies that would attack our own cells and tissues. Important also here, I just thought this picture illustrated it quite well. It's a pretty simple concept, but I do want you guys to know that your B cells are one of a kind. They are kind of like snowflakes in that sense. And a single B cell will produce all the different types of antibodies within the same variable region. However, really important here, B cells are extremely specific. Each B cell will make a unique antibody. So they are very, very specific. They're unique, but they're also very specific, sort of in the name there. So that's where we get to this point where each plasma B cell will make antibodies for only one antigen. Thus, for any given pathogen, there may only be a few B cells will be able to recognize it. So again, that specificity is inherent to the B cells then. So now we're going to be moving on to clonal selection of antibody-producing cells. So we will be discussing this process in the following two slides. And the process comprises of the following major occurrences, just to give you an overview, and then we'll be looking at pictures and all the like. So you will first note that there's inactive B cells that contain surface immunoglobulin that bind to an antigen. The B cell will then internalize and process the antigen. The antigen fragments will then be displayed on a major histocompatibility complex, MHC. Specifically for your B cells, this would be class 2 molecules. And then your T helper cells contracts the displayed antigen fragment and releases cytokines that activate your B cells. And then the cell will undergo proliferation. Just note on this major histocompatibility complex, I will point out, because there are two classes, I will be pointing out the differences. So this is what we were just on about. Here you will find that your B cell, you can see the B cell receptors on your B cell, and your extracellular antigens are present. So the B cell will then recognize and attach to the antigen. The antigen will then be internalized. You can see it's inside of the B cell. And the fragments of the antigen are then presented on the MHC. Again, this is your major histocompatibility complex, which is on the surface of the cell. So here you see specifically in this example, it's major histocompatibility class 2. that's then displayed on the surface. Following that, we will then move a T helper cell. We'll then recognize that this antigen fragment is activated and release cytokines activating the B cell. So here's your T helper cell that recognizes the MHC, and it will then release cytokines which will then activate the B cell. And then the activated B cell will begin clonal expansion, producing an antibody, producing plasma cells and memory cells. Very important. So that's why our subsequent responses will be much more effective. So here you can see the B cell with the antibodies, and B cells build bountiful antibodies. Just a little fun alliteration, which is just how these things function. So this is just an overview. Don't freak out that I cut some things off. I just wanted to show you what we will be discussing in the next couple of slides. So this is an overview of our antigen-antibody binding, which is the binding of antibodies to antigens to form an antigen-antibody complex that tags foreign cells and molecules for destructions by phagocytes and complement. We will be looking at each of these individually now. So first, there's agglutination. Which is essentially where antibodies cause antigens to clump together. For example, the two antigen binding sites for an IgG antibody can combine with the epitopes of two different foreign cells, aggregating the cells into clumps that are more easily ingested by phagocytes. So again, this is a very complex way of essentially just saying that it's kind of like sweeping dust into a pile before you put them in a dustpan. So you're just accumulating all these cells into little clumps or clusters before they become ingested by the phagocytes. So the point here is just to reduce the number of infection units to be dealt with, so that the little phagocyte doesn't have to move everywhere to pick up one. They're all pulled together, so it can just eat all of them essentially. Okay, and then we're looking at opsonation. Opsonation stems from the Greek word opsonare, meaning cater. The antigen, such as a bacterium, is coated with antibodies that enhance its ingestion and lysis by phagocytic cells. So again, that cater, think like catering or food, it's being made more delectable, if you will, for the phagocyte or the phagocytic cells. So essentially here, the point is to enhance phagocytosis through the coating, by coating an antigen with antibodies. So you can see here that they are coated, and the phagocyte can more easily or more readily ingest it. And then we have neutralization. So essentially in neutralization, IgG antibodies inactivate microbes by blocking their attachment to host cells, and then they neutralize the toxins in a very similar manner. So this will essentially make the cell or virus or toxin less damaging to the host. So you can see they've binded to the virus or to the bacterium, obviously probably not to scale. And this will block the adhesion of the bacteria or virus to the mucosa, so they can't attach, meaning, again, we've been through, I hope you recall from our pathogenicity lectures, that attachment is one of the primary things that most microbes need to do in order to cause harm. And similarly, if you have a toxin, they will bind to this toxin and make it unable to attach or then cause harm subsequently. So then we're moving on to antibody-dependent cell-mediated cytotoxicity. It's a mouthful, so bear with me here for a bit. So this antibody-dependent cell-mediated cytotoxicity essentially resembles opsonation in that the target organism becomes coated with antibodies. So there's a coating with these antibodies just surrounding the molecule. However, cell. I misspoke there. However, destruction of the target cell is by the immune system cells that remain external to the target cells. So this will be specifically perforin and lytic enzymes that help to destroy the cells. So they will they will destroy cells, and then phagocytosis can happen afterwards. So essentially here you have antibodies attached to a target cell causing destruction by macrophages, eosinophils, and NK cells. These are natural killer cells. And this is also mediated by attachments similar to, remember, opsonation, where you coat your invader cell. This is a very similar process to that. So then obviously we'll need to talk about the activation of complement. So this will cause inflammation and cell lysis. And here you can see your bacteria and your molecules, and then your complement over there. So essentially here, your IgG or IgM antibodies may trigger the activation of the complement system. So for example, inflammation is caused by infection or tissue injury. One aspect of inflammation is that it will often cause microbes in the inflamed area to become coated with certain proteins. This in turn leads to the attachment to the microbe of an antibody complement complex, and this complex lyses the microbe, which then attracts phagocytes and other defensive immune system cells to the area. So in this example that we have over here, the complement system is stimulating cytolysis using the membrane-attached complex. See, so here you can see the complement being attracted, and now the organism lyses, leaking its contents, which will then draw the phagocytes and the other immune system cells to the area. So now we're going to be discussing a bit of immunologic memory. Again, remember our primary versus secondary response, how much more effective the secondary, effective in terms of both reaction and recovery time, much quicker for your secondary response than primary. And that's due to your memory B cells, which will lead to an anamnestic response. So that's essentially a reaction, an immune reaction, where a bodily defense reaction that recognizes an invading substance, so an antigen, such as a virus or fungus or bacteria or transplanted organ, in a very less optimal example, and will then produce antibodies specific against that antigen. Again, here I decided to include the example of a transplanted organ because this isn't always a good thing, and that's why organ donors have to go through such a host of treatments where they have to suppress their own immune system at times to make sure that the organ that they received is not rejected by the body, because that is what can happen in this case, which obviously is not the desired outcome in those examples. Here we have an overview of the clonal selection and differentiation of B cells. So first we have stem cells differentiate into mature B cells, each bearing a surface immunoglobulins against a specific antigen. And then your B cell 2 encounters a specific antigen and proliferates. So here the antigen comes in, and this cell recognizes it, and then it'll start multiplying, proliferating. And then some B cells will then proliferate into long-lived memory cells, which at a later date can be stimulated to become antibody-producing plasma cells. So these will be your memory cells. This is what drives your secondary response and every subsequent response to be much more efficient. While other will just polymerate into antibody-producing plasma cells. This will actually fight off their primary infection, because these plasma cells will then be secreting antibodies into circulation. Here it is again, I would like to remind you guys that B cells can recognize an almost infinite number of antigens, but each particular cell only recognizes one type of antigen. So an encounter with a particular antigen will trigger the proliferation of a cell that is specific to that antigen. So this antigen that comes in here will only lead to the proliferation of this B cell, not other B cells. This will essentially lead to clones of this B cell being produced during the proliferation, hence why this is called clonal selection. So the initial antibodies, would also like to point out that the initial antibodies produced by IgM the initial antibodies produced are generally IgM, but later the same cell might produce different classes of antibodies such as IgG or IgE, which is called class switching, just a term I want you guys to be aware of. And yeah, now we'll be moving on here. Again, here you can see the primary and secondary immune responses to an antigen. IgM appears first, as you can see over there, in response to the initial exposure, and then IgG will then follow, and IgG, sorry, will then follow after IgM and provides longer term immunity. And then your second exposure, if you can see here, to the same antigen stimulates the memory cells, which was formed during your initial exposure, to rapidly produce a large amount of an antibody. The antibodies produced in response to this second exposure are mostly IgG, again represented by the black line. And you can also just see here, I cut off the figure a little bit, much to my annoyance, I just saw, but you can see just how many more cells are produced this time. So the response during your second exposure is much stronger than during your first one. And here, just to give you guys an idea, because the days are numbered pretty terribly down here. So your IgG will typically peak between like 10 to 17 days during the initial exposure, but during your second exposure, it'll peak between 4 and 7 days. So it's much quicker and you have a much greater response. Thus, you won't be sick as long. Okay, so now we're also going to be talking about types of immunity, and there are specifically two types of immunity, active and passive. And then we also need to talk about adaptive immunity, which is what we were talking about the entire time, actually. This refers to the protection an animal develops against certain specific microbes or foreign substances. And this immunity is placed into two categories, both active and passive. Both actively acquired immunity and passively acquired immunity can be obtained by natural or artificial means. So essentially, during active, we necessitate contact with the antigen for a long duration. This is essentially, you can also think of active as the natural process because it requires actual contact with the pathogen, but it can also be artificial, where we use vaccines or toxoids to produce kind of a immune response in when you predict that someone might be exposed to pathogen. Very similar to what's happening with the COVID vaccines now, where we're trying to make people get that secondary immune response so they don't become as sick. That's the entire point of the vaccine, is you want to expose people to a controlled vaccine. Thus, when if they then contract the actual COVID virus, it will trigger a secondary immune response, not an initial one. So they won't be as sick as long, and they will have a much greater response because of the Igs that are produced. So then passively, you can also transfer antibodies from one person to another. This will provide immediate protection, but it is temporary, unfortunately. This can happen naturally again here, where you have a placental or colostrum transfer from a mother, but it can also be artificial, where you just inject anti-serum or antitoxin from an immune individual to a susceptible person. Just for those of you who don't know, colostrum is a form of milk which is produced by the mammary glands of mammals, including many humans during pregnancy. And this colostrum will contain antibodies to protect newborns against disease, just in case you guys weren't aware. Okay, so this is just, again, a nice little quick overview of adaptive immunity, the difference between naturally acquired and artificially acquired. You can also watch a video here. This video specifically talks about antivenom, which is just something that you guys might not find on your own. So I just thought that's also helpful. Very helpful for exams. And that is essentially it, guys. So let's do some review questions today. Just a quick little true and false here. True or false, immunological tolerance is the failure to mount an immune response to an antigen. I'm going to give you guys a couple of seconds. Pause the video if you need some more time. So the answer here is true. Moving on to epitomes are small molecules that elicit an immune response only when attached to large carriers such as a protein. Is this true or false? Is that what epitomes are? That's false. Remember, the correct answer is a hapten. A hapten are the small molecules. Okay, that's a typo over there. B cells are not specific. Is this true or false? I mean, this is pretty simple. I kind of stressed this quite a lot. So I'm going to move on here. The answer, of course, here is false. Remember, they are in fact very, very specific because each B cell makes a unique antibody. And then question 4, B cells will attack invaders inside of cells. And the answer, of course, here is false. T cells will attack inside cells, and we will be discussing them in our next lecture. Okay, guys, thank you so much, and see you in the next one.